is an essential version of food that has been given to fish fry for many ages. It is scientifically proven that it can result in high productivity of fish fry with a low chance of fish mortality. Some species that usually cultured in large scale are microalgae, rotifer, and microworms. There are many advantages of giving this type of feed to the fry in order to increase the rate of productivity and effectiveness at the optimal level in culturing fish. Microalgae refer to microscopic plants that can be found in both freshwater and marine systems. There are unicellular species which can exist individually or in groups. There are 4 basic categories of culturing methods that have been used in culturing this feed. The first is indoor or outdoor technique. Some fish farmers prefer indoor culture method instead of the outdoor, since there are many benefits in using this type of method. Next, it can be classified as open or closed technique. After that, azonic or zonic technique, which refer to the condition of the environment either free from any types of contamination or the condition with the presence of bacteria. Lastly, culture of the media either in batch, continuous or semi-continuous, which refer to the skills of culturing the live feed. Microalgae and periodic cell size for ingestion, which in range 1 to 15 micrometer and can reach to 100 micrometer. It contains high level in vitamins such as vitamin A and vitamin B. Microalgae is rich with good nutrient composition and does not produce toxins. There are some important parameters that need to control in culturing microalgae. Nutrient quality and quantity are very important to take care as they are the source of the presence of microalgae. As for light, it is the source of energy that leads to the photosynthesis process. The range of most suitable pH is 7 to 9. Aeration and mixing the mixture is necessary to prevent the sedimentation. The best temperature for this type of live feed is between 20 to 24 degrees Celsius, with the optimum level of salinity is between 20 to 24 pt. There are 5 phases for growth of microalgae culture. The simplest method to culture microalgae is as follows. Add aquarium cell into a gallon of fresh water and stir the water. Then 40 drops of fertilizer was added into the water gallon and left for overnight. An aeration flow was added into the mixture to mix them. Then the culture should continue with a new culture to continuously supply for this live feed. The constraints are relatable to microalgae culture. These are the supplies that are very costly. It also requires special facilities and space. In microalgae counting, in about 5 ml of sample taken from each culture and placed in a separated test tube, then add 1 drop of Lugo solution. A drop of well-mixed algal suspension was put into the chamber and covered with the cover slips. These are the calculation method they use to count the cells. We should done this to evaluate the production of cell growth and to regulate the population of the microalgae. Rotifer. Rotifer is zooplanktonic type of multicellular animals which have organs for locomotion, feeding, digestion, and reproduction. They are usually very small in size, which range in between of 200 to 500 microns. It is suitable for fish larvae. Cultivation of rotifer can be performed by using this method. Cut 12 inch of rigid tubing, then attach the bottle to a valve that connects to an air pump using a flexible tubing. The bottle was emptied before it filled with starter rotifer culture. After it is become high in concentration, collect them to fry and store the rest in the refrigerator. The advantages of this culture: it is smaller than Artemionopolis size, tolerance to wide range of environmental conditions, high reproduction rate, and slow swimming velocity, which make it easy to prey by fish fry. Microworms. Microworm is a free-living type of nematodes which is very suitable to feed to fish fry. The steps of culturing this live feed are: put an inch of cooked oatmeal layer at the bottom of a container. Then 1 teaspoon of dry yeast was added into the oatmeal before left for overnight. After that, the microworm starter was added into the mixture. The culture was stirred once a week to keep them alive. In the conclusion, every type of live feed have different types of nutritional value. This includes the artificial and the live feed. The fish farmer should choose the best type of food which can bring an optimal rate of productivity.